Now, so let's do a very quick review of what we have done so far. So what we have done so far is essentially going from glucose, and we go down using glycolysis. And this takes you to, for example, PEP. PEP goes to pyruvate. Pyruvate goes to acetyl-CoA. And of course, we saw that PEP and pyruvate, these guys can come down to oxaloacetate. And this whole thing is called the TCA cycle. And as a net product of TCA cycle, you have 2 CO2, 3 NADH, 1 FADH2, and 1 ATP. Okay, and of course here you also produce ATP, here you produce NADH, etc. And here you also produce ATP, NADH, things like that. Okay, so we talked extensively about glycolysis, and of course we also have going from pyruvate essentially back, gluconeogenesis. Okay, and we also talked about glycolysis can do a bypass. This is the PPP generating NADPH. And we also talked about a little thing here called the glyoxylase shot. This is the glyoxylase shot. Okay, things like that. Okay, so you should have a pretty good idea of essentially how the energy is transformed from glucose to CO2, and in the process you get, you know, how many ATPs, how many NADH, etc. Okay, so that was sort of the what we generally call central metabolism. Okay, so this is what we generally call central metabolism, and this is what happens to your bread that you're eating. You know, you digest your bread, bread becomes glucose, glucose essentially becomes CO2. Okay, and of course, as metabolism go, okay, later we'll talk about amino acid biosynthesis. So for example, PEP can go to amino acid, OA can go to amino acid. What else? For example, here you have alpha KG can go to amino acids. So a lot of the stuff, a lot of the key central metabolites that you produce can also go to amino acids. Okay, so we'll talk about that later. Okay, so really from today on, okay, from today, starting today, our focus will be more on the anabolic pathways. So more about how to make biological components. Okay, so previous lectures were really about how to generate energy from biological components like glucose. Okay, so from now we're going to be talking more about how to build biological parts. Okay, of course we'll still talk about degradation. For example, when we talk about fatty acid degradation, we'll still talk about how to get energy out of fatty acids. But you know, keep in mind that nowadays we're going to be talking about biosynthesis quite a bit. Okay, so as you can see from, you know, the topic, we're going to be talking about photosynthesis. Okay, now photosynthesis, really, I mean, obviously you should be able to appreciate the importance of photosynthesis. I mean, no shit, because most of the energy we get from outside is essentially coming from the sun. But you know, nonetheless, photosynthesis is a very important topic for biochemistry. Now, it's used to provide carbohydrates, okay, for energy production, and this energy production is referring to us. Like when we need to generate energy, a lot of times we use carbohydrates to produce our energy. But of course, carbohydrates come from somewhere, okay? And the source, okay, is essentially photosynthesis, right? And the key for photosynthesis is that it's going to fix CO2, okay? So basically, we're going to use the energy of the sunlight and essentially store that photo energy into chemical energy stored in organic products. So what we're going to do, okay, is converting CO2 into central metabolites. Okay, so that is photosynthesis. So the key is converting CO2 to central metabolite. Okay? Now, for us humans, like animals or E. coli, what we call ourselves we are heterotrophic, okay? Meaning that we get our energy and carbon from somewhere, okay? From organic carbons. Okay, so our carbon source, organic carbons like glucose. Our energy source, organic carbon like glucose, okay? So we are heterotrophic. And essentially, heterotrophic, we take carbohydrate, okay? So this is the carbon, energy, and electron source for us, okay? And we have molecular oxygen that will help us to oxidize these organic carbons such that you can release that energy. Okay? It's the same thing as you take a bunch of wood and you light it on fire, right? So in order to have energy release, you need to like combust something. Very similar idea. Okay, you are oxidizing organic compounds, and as a result, you release energy. Okay, and of course, through respiration, you will get CO2, water. Okay. And autotrophic cells, obviously, then will then be able to use the energy. So this guy is the energy that will convert this carbon source, water, electron source, okay, and grow on CO2 and light and water. Okay, so they can then convert these two things plus solar energy, and we'll convert that into carbohydrates. Okay, and then we can then use it again. All right. So today, my primary goal is to talk about photosynthesis, the light reaction. We'll talk about how photosynthesis can be split into light reaction and dark reaction. And the light reaction, just keep in mind, is pretty much the reverse of respiration, okay, or the reverse of ETC. Okay, but it's also a kind of electron transport chain. Okay, now one of my goals for you is as you're learning metabolism and as you're learning biochemistry, you really should be able to answer the source of these three things for any organism. Okay, any organism on Earth requires the source of carbon, source of energy, and source of electron to grow. Okay, all organisms. Now, again, for animals or heterotrophic cells like E. coli, carbon, energy, electron, they're all organic carbons. Okay, organic carbon meaning like glucose or organic acids, that kind of stuff, right? And for plants, the carbon source is CO2, energy source is the sunlight, electron source is water. Okay, of course, we'll see some other interesting organisms that will have different carbon, energy, and electron source. Okay, all right. So this is just carbon cycle in nature. Okay, just to tell you that Earth as we have it right now. Okay, Earth. Obviously, everything has to be balanced. Okay, so one of the things that you should understand is that anything you produce, okay, anything that biological cells produce, there's going to be something else that can consume it. Okay， 听懂我意思吗？就是你一个 cell， 如果你生产了一个什么东西，一定会有别的 cell 可以去吃它。OK， 不然的话，你可以去想一件事情，就是 like， 如果有一个 organism 它 net produce 一个 chemical，OK， 那没有别的 organism 可以去吃这个 chemical 的话，你会有一个 net accumulation，right？ 那如果有这个 net accumulation 的话，你地球就是不断的会增加这个东西嘛 ，right？ 不合理啊，懂我意思哈？ 就不太合理啊，因为 otherwise 我们就应该要看到，但是没有 ，OK， 所以地球是一个balance 的 process 这样 ，OK，so basically carbon cycle is that CO2 is fixed by autotrophs, so photosynthetic cells, and then they generate carbohydrates or lipids, whatever, and then there are the heterotrophs that can take those things and then use them, take out the energy from organic carbon, and we use respiration. Okay? Right? So photosynthesis, okay, again, it requires a reductant source of electrons. Okay? Source of electrons. Why is that? Because if you look at CO2, okay, CO2, the carbon here is very oxidized. Right, because it's attached to essentially four bonds to oxygen. So in order to store energy onto CO2, you have to reduce it, okay? And if you're talking about reducing CO2, then you're talking about you have to have a source of electron in order to reduce your CO2 and make it into something, okay? So what you need to do is you're trying to reduce CO2, okay? Like reduce 不减少还原的意思 ，OK？ 那英文有时候就是怪怪。All right. Now, of course, typically for most photosynthetic organisms, we're talking about water, okay? Because if you're talking about water, water is essentially you can see it like that, okay? Water is essentially like that. If you balance it, it's like that. And then hydrogen, this is electron, okay? Inside of hydrogen gas, you have electrons, right? So, but of course, what we're going to talk about today is really how to split water, okay? So your electron source doesn't have to be water, you know, as long as you have some kind of substance that has electrons right here, you can use that as the electron source, okay? So there are a lot of different organisms on Earth that can take other things for source of electrons. Now, we are most comfortable and most familiar with cyanobacteria. This guy is like, you know, plants, algae. Those things are similar to cyanobacteria, where the reducing power or the source of electron is water. Oxidize the product, of course, water becomes O2, and the source of carbon is CO2. Okay? That's simple, like your plant, like your trees. Now, there are other bacteria around that can do photosynthesis, okay, and can do autotrophic growth. Okay. By the way, when I say autotrophic, do you guys know what that means? 知道吗？就是可以用 CO2 长，可以用 CO2 长就叫 autotrophic， 就是 auto 嘛，就是有点像那种自动的意思，所以它可以自己长。OK， 然后 heterotrophic 就是说它是需要吃其他的 organic compound 来长。Okay, so heterotroph versus autotroph. Okay, now there are quite many other types of bacteria that can use different electron source. For example, H2S, hydrogen sulfide. Okay, so if you take out the electrons from hydrogen sulfide, you can oxidize the sulfur right here, and you turn it into sulfate. Okay, so hydrogen sulfide becoming sulfate, and the electrons are taken out and stored on CO2. Okay, stored on CO2. Right, and then it can grow. There are other bacteria. Oh yeah, by the way, these things are called sulfur bacteria because they use hydrogen sulfide as the electron source. That's why they're called sulfur bacteria. Don't ask me why they're purple and green. I'm just kidding. If you're interested, you can come ask me anytime you want. And you also have these purple non-sulfur bacteria. They are also photosynthetic. Right here, the chart here is photosynthetic. However, what you notice is that the source of electron can be hydrogen or it can be reduced organics. Okay, so that means that this guy can take in organics. Okay, so it can also use glucose or acids and that kind of stuff. Okay, And the oxidized product, we're just going to call it generally oxidized products. Okay, so you can turn reduced organics to oxidized organics. Okay, and source of carbon can be CO2 or organics. Okay, so basically this type of organism can be both autotrophic or heterotrophic. Organisms that can do both, sometimes we call them mixotrophic. Easy enough to understand. Mixotrophic. Okay, 就是 maybe 两个都可以做 simple. Right? And you also have this other type of bacteria called the heliobacteria, and this guy is just kind of crazy because it doesn't really grow on CO2. You see? Okay, it doesn't grow on CO2. Basically, it's nothing different from like a heterotrophic bacteria. Okay, so the source of electron is going to be organics. Source of carbon, organics. All right. So basically, what happens is that it can use photosynthesis to help it grow. Okay, but it is required to do heterotrophic growth, meaning that it's like E. coli, but you can use photosynthesis to help you grow. Okay, but you are absolutely required to use heterotrophic growth. You are required to use organic carbon to grow. Okay, but cyanobacteria, green sulfur bacteria, purple sulfur bacteria, they don't require that. They can do complete autotrophic growth. Make sense? Right? Okay, so this is the overall metabolic map that we keep on seeing. This is from your book. So we talked about the stuff down in the middle. Okay, so we talked about essentially this part. Okay, so this class, next class, maybe next class, we're going to be talking about the green stuff. Okay, you know, this thing, the green stuff. Okay, I like how it's green because you know it's green, right? So yeah, we're going to be talking about photosynthesis, and if you see, you know, this scheme, essentially you already know what photosynthesis does, right? It's going to capture CO2 and go through this process called photosynthesis. You're going to return glyceraldehyde 3-phosphate. Okay? So your net reaction is going to be CO2 becoming glyceraldehyde 3-phosphate, G3P. So, you know, that's how it's done. And so for algae or for plants, you know, when they grow, they are going to grow on CO2, make G3P, and once you have G3P, you have everything else, right? G3P can make glucose, G3P can make pyruvate, pyruvate make acetyl-CoA, acetyl-CoA make fatty acids and that kind of stuff, okay? As long as you have G3P, you can make everything else, okay? G3P is just a 3-carbon sugar. Glucose is a 6-carbon sugar. All right. So photosynthesis can be split into 2 parts. Okay, there's the light reaction and the dark reaction. Okay, the dark reaction is a lot of times called the carbon assimilation reactions or carbon fixing reactions. CO2 fixing reactions. Okay, and essentially you can split the entire photosynthesis into 2. Okay. Now, the light reaction, okay, as the name suggests, it has to do with light, okay? And what it does is really, the light reaction is you are going to use this energy right here and take the electron, okay, electron from water, dump it onto NADPH, okay? In the process, you also generate ATP. Okay, so light reaction basically use visible light as the energy source, and it produces reducing power, and reducing power is coming from electron from water, okay? And you generate NADPH and ATP, okay? O2, this guy is a byproduct, okay? And then in the dark reaction, okay, in the dark reaction, the NADPH and the ATP are then going to be used to fix CO2. Okay, remember we said that CO2 plus electron can then become central metabolite. In this case, G3P. Okay, and this electron is NADPH. Of course, you also need to use a little bit of ATP to help it work. Okay? Now, here's a question for you guys. Does the dark reaction have to happen together with the light reaction? 你可不可以把 light reaction 跟 dark reaction 完全分开？ Maybe. 就是 chemically speaking, you can, 对不对？就是 dark reaction， 反正它是吃 NADPH 跟 ATP 嘛 ，right？ 那 light reaction 是 generate NADPH 跟 ATP 嘛 ，right？ 所以其实我们站在一个 chemistry 的角度的话，你可以说呀，因为 as long as 我这个 cell 里面有 NADPH 跟 ATP 的话，我还是可以做我的 dark reaction， 就是我还是可以 fix 我的 CO2，right？ 那 light reaction 就是我只要有水跟这个阳光，我就还是可以做得到我的 NADPH 跟 ATP，right？ So chemically speaking， 它们两个是可以被分开的 ，OK？ 但是 turns out for most organism，OK？For most organism, light reaction is coupled to the dark reaction. Okay, a lot of times this has to do with regulation. Okay, it's actually on the regulation level that they are related. For example, cyanobacteria, it cannot live without light. Okay, 这其实很 interesting 哦，因为 cyanobacteria 其实你只要想说我们 as long as we give it like a source of electron and energy, it should be able to grow. But it turns out it does not grow. Okay, you have to. People have done this experiment where cyanobacteria is photosynthetic bacteria. So if you want it to not die, you have to minimum 1 day. You have to give it like 15 minutes of light. Okay, so it's actually regulation, right? Because without it, it cannot live. Right. So light reaction and the dark reaction. Right, so let's start from something very simple. Photosynthetic prokaryotes, these things are called cyanobacteria. Cyano is like blue, right? Cyan is blue, so it's the it has another name called the blue-green algae. But not algae. OK， 你要是去那个 Wikipedia， 你打 cyanobacteria， 然后你去那个左下方按那个中文有没有？它会出现蓝绿藻 ，OK， 但是它 technically speaking， 它不是藻类，因为藻类通常是这个 eukaryotic cells，OK， 它是 prokaryotic cells， 哎，所以很多人有一些那个很坚持的那种微生物学家哦，就是那种 microbiology， 他们会说这不叫蓝绿藻，这叫蓝绿菌。whatever 啊 ，whatever。那它为什么会叫蓝绿呢？主要是因为它里面有很多那个 protein， 专门帮忙吸收光的 protein，OK， 是蓝色的。Okay， 但是蓝绿菌，虽然它叫蓝绿哦，但是你看眼睛上看起来是绿的哦，它不会来变蓝色哦，它 still green 哦。Alright, so photosynthetic prokaryotes, cyanobacteria, for example, turns out. What you can notice from this picture right here, what you should see is that they have these crazy membranes, right? And that is because your photosynthetic machineries, the light reaction machineries, all happen on the membrane, okay? They're all on the membrane, okay? Just like what? Just like your electron transport chain, okay? So all of these machineries are on the membrane, okay? And obviously, if the proteins are on the membrane, you should have more membranes, so in order to do your photosynthesis, okay? So you have a little cartoon here, okay? You can see it right here. We have this thing called photosystem 1, cytochrome B6F complex, photosystem 2, okay? And then we have all these proteins right here, okay? So all these proteins right here. And because this is like a little extra layer of membranes, you will have a separation of space, okay? So you have this thing called stroma, and you have this thing called lumen. Just like your ETC, lumen, this guy is the intermembrane space. Yo. Okay? Right? And then this guy for mitochondria is like the matrix. 我放括哦 Okay, just like quote. Right. Yeah, well, whatever. Right. So the lumen is just like the intermembrane space for mitochondria. Okay, and the stroma is just like the matrix of mitochondria. All right, but for your purpose, lumen, stroma, intermembrane space, matrix. Okay, just like that. All right. So if we're talking about eukaryotes, like plants and algae, like the real algae now, eukaryotes have this thing called chloroplast. Right? Chloroplast 的中文是什么我也不知道，我也不是很 care. Right? But chloroplast, the thing that has chlorophyll inside, that does photosynthesis, 跟你打赌已经是绿 something 的，中文叫什么？叶绿体嘛，对，我就跟你说有绿嘛，好不好？ Alright, so chloroplast, it's just like a cyanobacteria. Okay, so inside of your leaf, you have these organelles called chloroplast, and chloroplast is in a way kind of similar to a cyanobacteria. Why? Because you have a outside membrane, and then inside you have these what we call thycoid membranes. Okay, all right. So by the way, these things, they're called the thycoid membranes. Okay, thycoid membranes. Right, because the cell membrane is like right here. Those are the cell membrane. Okay. So these are they're called thycoid membranes. So inside of your chloroplast, you also have these thycoids. Okay, so they call the entire thing thycoids. Okay, but you just understand that inside. So inside here, you have the lumen. Okay. All right. So yeah, whatever. So this guy is just like a little cyanobacteria. Okay. So as long as you understand this thing, right? This chloroplast, it's just like mitochondria. Okay. Except that the enzymes are different. Okay. For your purpose. Right. So like a plant cell, this guy is a plant cell. Plant cell have cell wall. The last time I saw a plant cell was like in 国中吧, middle school. Right, so you have mitochondria, and then you have these things called chloroplast. So, and inside the chloroplast, you have these thycoid membranes. Okay? All right. So that's the physiology or the pictures of biology for you. Now, let's talk a little bit about energy, right? Because we're still interested about energy, right? Because photosynthesis is really a energy conversion. We're converting electromagnetic wave, EM wave, into chemical bonds. Okay, right? So we're going to sort of transfer that energy. So let's very briefly take a look at how much energy is available, okay, to do this. So now recall that energy of any EM wave is energy is equal to H times nu. Okay, well, whatever. Okay, you guys know this from like high school, right? But we know that if we want to know the energy of 1 mole of light, okay, and again, light is both a wave and a particle. Yes? 有了哈. Any EM wave, okay, EM wave, light, EM wave, same thing, okay? So any EM wave, you can treat that as a particle or a wave, okay? And let's say if we want to look at a energy in the particle sense, sort of, okay, we can sort of calculate the energy associated with a 650 nanometer light wavelength. So this guy is typically in the red, right? So this guy is red light, red or orangey light. 700 is red, but it's towards red. Okay, and that will give you an energy of around 184 kilojoule per mole. Okay, because this is the energy per particle. Okay, this is energy per particle. And of course you times it by, what's this number called? Avogadro's number. I can't spell his name. Starts with an A and V. something. Okay, Avogadro's number. 不是 avocado, avocado 是吃的哦 Avogadro 是 the guy's name 哦 Okay. All right, so if you are looking at a 650 nanometer length of light, that gives you about 184 kilojoules per mole. Okay, and basically all photosynthesis is really occurring using visible light. Okay, so IR, most of IR is not, the energy is not high enough to do photosynthesis. UV, a lot of it is blocked by our atmosphere, okay, and energy is too high, right? So turns out biological photosynthesis is really through evolution optimized to use the visible light spectrum, and obviously you can see how narrow this thing is. It's a very narrow gap, right? So when we look at energy of photons and compare that to energy of chemical bonds, okay, this graph essentially shows you energy per mole of photon as a function of wavelength. Okay, so this is wavelength, and this gives you energy. And then compare with the energy of several chemical bonds. And you can see how in the IR you have carbon-fluorine bonds, carbon-CH bonds, CC bonds. So these bonds, high energy. Okay, so obviously we are not going to use light to directly, break or make mess with these bonds. Okay, so we don't do that. We are concerned with the visible spectrum. Okay, so visible light energy is of course lower, and if we are going to translate this energy to chemical bonds, you are looking at carbon iodide bonds or carbon nitric oxide, so NO bonds. This is just to show you where the energy is. Okay, it's not really relevant to what we're going to talk about. Okay, but as long as you understand that around this wavelength, that's where the energy of chemical bonds are stored at. Okay, so chemical bonds turns out 就是差不多在 UV 跟 visible range. Okay, 不是什么 X-ray 啊，也不是什么 like a radio wave 啊 ，right？ 你看我们的整个EM wave 它 spectrum 很大，那 chemical bonds happen to be around here. At least the organic chemical bonds. Energy is around this region. Okay? All right. So we're going to talk about how light is absorbed, right? Okay? Alternating double bonds are called what? 这是一个 question 哦。Conjugation. Okay, so anytime when you have a conjugation inside a molecule, this molecule typically will have colors. Okay? So all of these molecules have colors. Right? Now, chlorophyll A and chlorophyll B, okay, chlorophyll is probably the most abundant plant and algal pigment. Okay, so that's why these things look green. That's because chlorophyll looks green, right? And chlorophyll A and chlorophyll B essentially looks like this. Okay, so this guy is chlorophyll. Okay, and obviously you see that it's very similar to what? Heme. Okay, it's very similar to the heme that we previously saw, okay, like hemoglobin, like cytochrome, that kind of stuff, okay? But what you should notice here inside it's a magnesium atom instead of iron, all right? But regardless, because of the conjugation right here, okay, it's going to be able to absorb light, okay? Right? And we have other types of photosynthetic pigments. These are called photosynthetic pigments. Pigments are, color molecules. And these things are like phycocyanin or the phycoerythrin. Okay, these are open chain tetraperols. So they're, also, this thing is a perol and open chain. So it's like this, but not a big circle. Okay, but whatever. Okay. They absorb light. Okay, they absorb light. Okay, and phycocyanin especially, it's more or less important for me, at least, because they are abundant in aquatic photosynthetic organisms like cyanobacteria and algae. Okay, so different photosynthetic organisms have different amounts of different pigments, obviously. You also have things like beta carotene that has extended conjugation system. Oh, phycocyanin is right here. 就是这一个东西啊。叫做 perol. 然后 both chlorophyll and phycocyanin, they are tetraperol because they have 4 of these rings right? But chlorophyll, they are connected into a bigger ring, but phycocyanin is not, okay? It's actually bound to a protein. Whatever. Regardless, these pigments will absorb strongly in 500 to 600 nanometers of a range. I'm talking about these things, okay? Because those waves can penetrate water pretty easily. So that's one of the reasons why aquatic photosynthetic organisms will actually have more of phycocyanin type of molecules. Okay? We'll talk a little bit about bacterial chlorophyll later. Okay, so as long as you understand that these molecules with conjugations will absorb light, okay, which is why if you have a bunch of those on the table, they look colored. Okay? Color meaning they absorb light. Now, so this is a very interesting graph that I really like. This is comparing natural sunlight, which is the black line. Okay, the black line is sunlight. Okay, is the light hitting the surface of the Earth. Okay, and obviously you have a bunch of IR. So here, IR. Okay, and below here, that's going to UV. Okay. And we see that a lot, okay, about I think more than 50% of the light, okay, hitting the surface of the Earth is in the visible region, okay? And obviously we call sunlight white light because it has every wavelength, so it's white. Now, obviously what you can notice is that different pigments, different photosynthetic pigments absorb with the peak at different wavelengths. Okay, so different pigments absorb at different wavelengths. Now, the question then I ask you, okay, let's say your plants or your algae have all of these pigments. How come your leaf is still green? How come it's not black? If you have all these pigments, how come it's not black? 有没有想过这个问题？就是如果说天然界里面有这么多不同的 pigments 可以去吸收光，那好啦，那你为什么不是就可能一个植物，然后里面这些 pigments 都有，然后全部的光都吸？那如果全部的光都吸的话，这个东西会看起来像是会黑色嘛？对，黑色就是代表它全部都吸。那你的植物为什么不是黑的？绿色比较多。啊？吸绿色，应该说它不吸绿色的东西比较多，对不对 ？Okay, that's exactly right. Right. So the reason why your plant leaf is green because, like I said, plants have a lot of chlorophyll. Okay, chlorophyll is the major. Okay, is the major photosynthetic pigments in most of organisms that do oxygenic photosynthesis. Okay, so it's a lot. Okay, primarily it's chlorophyll. Now look at where chlorophyll absorbs. You have one absorbing at about 400 and 450. That's one peak. Another peak absorbing at the red. Okay. And then chlorophyll B, same thing. Here and here. 我这样画一圈，然后你也看不出来什么是什么，就是这个。Right, so those two are the chlorophyll. And obviously, chlorophyll have two characteristic peaks. Okay, so in terms of absorption, they absorb 400. They also absorb 700 here and here. Okay, so turns out, okay, really, it turns out chlorophyll absorb the blue light and the red light. Okay, but if you see here, this region, it doesn't really touch the green region. Okay? Makes sense? Right, so that's why your plant looks green. Alright. Whatever 啦，反正就对，好不好？ Okay. Now, let's talk about a little bit. Let's talk a little bit about this energy transfer mechanism. Okay? This is less biology, more chemistry. Okay. Now, we're going to have two types of transfer. One type is called the resonance transfer. The other type is called the electron transfer. Okay, the resonance transfer and the electron transfer. All right. The resonance transfer, okay, it happens between two identical molecules. Okay, so identical molecule. Basically, okay, let's look at resonance transfer. Okay, let's look at just here for now. You have two molecules, molecule 1 and molecule 2. They're same thing. Okay, now molecule 1 and molecule 2, they're both photosynthetic pigments. That means that they have conjugation. That means that they have a lot of pi electrons that can be excited. Okay, 那个各位 excitation 啊，就是一个 electron 被 excited 什么意思 ？Anyways, as long for your purpose, electrons have ground state and excited state. Okay, and how does it become excited? Typically, is through light. Okay, through some kind of photo excitation or some kind of energy input. Okay, now your electron, because your color molecules, they have these conjugation, and anytime you have conjugation, basically what conjugation does is it allows your electron to travel freely. 你知道吧 ？conjugation 这个东西，讲一下 benzene ring 好不好？我们讲一下 benzene。benzene 这种东西其实也算是一种 conjugation 一种啊，它是 aromatic， 对不对？那 benzene 的话，它的 orbital 长这样，它的 pi orbital 长这样。OK， 那每一个 carbon 身上都会有 pi， 就是都会有 pi bonding。OK，pi bonds，right？ Now pi bonds 它们的 bonding 方法是 through sideway contact， 对，它不是 head and head。这 head and head 的这个东西叫 sigma bond，right？ 那 pi bond 它是 through XYZ 轴的 interaction，OK， 它是 XYZ 轴的 interaction。那如果我们今天再看一个benzene ring 的话，你们知道 benzene ring 其实中间会有一道那个 magnetic wave。有机有没有讲过这个？因为当你这边所有的 electron 是通的，哎，所以你其实就是暂时可以，其实会有个 current。哎，如果你拿那个 NMR， 哎，你去弄它，你去造一个那个 wavelength， 它其实你可以 induce 出一个 current。那你 induce 出一个 current 的话，你有 current 就代表你有 B field， 对不对？你有那个 magnetic field， 它可以往下走。哎，那所以这只是要跟你讲说 conjugation 这种东西，就是当你今天有 conjugation 啊，那你看你的每个 molecule 有 conjugation 的时候，每一个 carbon 身上都会有它的 pi electrons， 就是都会有它的 pi orbital with electrons， 都有 pi bonding，OK， 都有 pi bond。那都有 pi bond 的时候，你的 electron 其实可以就是比较 disperse， 就它不是卡在一个 local 的地方，所以当它可以 freely move around 的时候，它的 energy 其实 typically 都比较低。OK， 那 electron energy 如果比较低的话，它就比较容易被 excite， 就是说它的不能讲说它的 energy 比较低，它的excited state， 它的 excited state 的 energy 会比较不会很高。Okay? 所以 typically conjugated systems can absorb visible light because you know when they absorb light, their electron gets excited. Okay? Right? But whereas let's say you have just alkene, like a random alkene, one alkene, okay, you can't see it, but UV can. Okay, you can detect alkene using ultraviolet spectrophotometer, right? You can detect that, but you know you cannot detect, you cannot see it in visible range. Okay, so that's because when you have conjugations, you have your electron is easier to be excited. Right? Now, so your molecule, you have one molecule, two, same thing, same molecule, and you have a photon coming in, a light wavelength coming in, and it's going to excite one electron on the molecule, okay, excite it into the excited state. Okay, so obviously this is energy input. Okay, photon is the energy input, and then you got an excited state. Okay, so here your electron is in the excited state. Now, resonance transfer, what it does is it's going to sort of going from molecule 1, and your excited state go from molecule 1 to molecule 2. Okay, so you pass that energy directly to an identical neighbor molecule such that you excite the electron on molecule 2 into the excited state, and then molecule 1's excited state comes back down. Okay, so energy is like translated. 你们有没有看过那个桌子上不是有一种那个 elastic collision 吗？你们在物理不是有学过那个 elastic collision， 就是那个球有没有？你这边拉一颗球这样弹过去，然后那个能量不是会传递到后面那颗球，然后那颗球不是会飞起来？ 类似啊，类似的概念，就是你的 energy transfer 从这颗球到它旁边那颗球，到旁边那颗球，到旁边那颗球 ，eventually 到另外一颗球，然后那颗球可以飞出去。Okay, so it's similar idea to elastic collision. So it's like energy conservation. Energy is conserved in the excited state, right? So you go from molecule 1 to molecule 2 without losing energy. Okay, so this is called the resonance transfer. Now, electron transfer. Electron transfer is something different, right? Where you have different molecules. You have different molecule 1 and molecule 2, okay? And molecule 2 has a electron state, okay? It can accept an electron. Okay? It could be like, I don't know, NADP becoming NADPH, for example, okay? It has it can take an electron, okay? But the electron state not necessarily have to be excited, but that electron, you can take an extra electron, basically. Now, the molecule 1, same thing. You excite it with a photon going into the excited state, okay? Now, what this guy can do is then it can do an electron transfer. Okay? So it can pass the electron onto molecule 2, okay? Onto molecule 2 such that your molecule 1 now is 1 electron short. Molecule 2 is 1 electron short. The opposite is 什么？不要跟我说 long 哦。Extra. Okay? So 1 extra electron. Okay? So by doing this, you achieved a separation of charge. Okay? So you passed an electron from your molecule to another molecule. Okay? Obviously, molecule 2, this does not have to be some like crazy stuff, but you can pass the electron onto another molecule. But this other molecule typically have a lower energy. Okay. Why does it have to be lower energy? Because otherwise delta G would be positive. Okay, so you want this process, delta G less than 0. Okay, this is electron transfer. So you transferred an electron, right? Now, why is electron transfer exciting? 我没有觉得很 exciting 啦，你可能也没有觉得很 exciting 啦。但是 electron transfer 就是可以做 electron transport chain 啊，对不对？可以做 NADPH 啊，就 not bad 啦，还不错啊，是有 function 的，好不好 ？Okay. So just to correct something I probably said wrong, molecule 2 will have a lower excited state, but that doesn't really matter. What really matters is that as long as you have another electron acceptor that can take in the electron, that will be okay. Okay, so this is sort of like electron transfer and resonance transfer. Right? Now, resonance transfer, okay, is used for light harvesting. Okay? By harvesting, we mean you harvest, right? You 收刮 light. Okay. Now, light harvesting is like when you on the what do you call this? Thyroid membrane. On the thyroid membrane, okay, you have a big protein complex here. This protein complex is called the photosystem. Okay, photosystem, okay, or photoreaction center, I should call. Okay, so this is the photoreaction center. So this is where photosynthesis occurs. So here is the key reaction that occurs. Okay, but it is not feasible to have a bunch of these proteins. Okay, it doesn't work that way. So it turns out most of these photosynthetic organisms, they will develop photosynthetic pigments around the photosynthetic center. Okay, so you have a bunch of other photosynthetic pigments. Okay, so kind of like this. So you have other photosynthetic pigments that are surrounding your reaction center. Okay, so this is your reaction center. Now, it's like you want to spread satellites. Okay, so these are kind of like satellites where you have photons coming in, hitting this molecule, and you can do resonance transfer. Okay, so the energy between these guys, they're all resonance transfer. Resonance transfer. Okay, so they keep on resonance transferring to different molecules until you hit a molecule that is next to the reaction center. Okay, and then you have a chance to pass that energy onto the reaction center. Okay? So once the energy is at the reaction center, okay, then it's going to be able to pass the electron to a electron acceptor. Okay, this could be ferredoxin or iron sulfur cluster, something. Okay, so as long as you can absorb light and keep on passing the light, okay, using resonance transfer and passing, eventually you pass to the reaction center. Okay, and once at the reaction center, you can then pass the electron to a downstream electron acceptor. Okay, so this is then electron transfer. Okay, so this guy is electron transfer. And then all these things are resonance transfer. Okay, so these pigments, we usually call them antenna. Okay, like a 天线有没有专门来吸收 light 的，吸收wavelength 的。Okay, 所以 antenna molecules, these are called. Okay, and typically you have a bunch of antenna molecules spread around, so that all of these antenna molecules, they can all absorb light and keep on passing the light very quickly to their neighbor, eventually to the reaction center, and the reaction center can then throw the electron over to the next acceptor. Okay. Obviously, okay, 如果你现在有在思考的时候，你会心想，哎，奇怪 ，electron 从哪里来？有没有想过这件事情啊？我 electron 就弹出去，有没有？那我那个 electron 我的那个 reaction center electron 不是越来越少？可以哦。当然不行嘛，对不对？没关系，让我们继续看下去，好不好 ？Okay. So the way it works is like this, okay? So this thing is called the Z-scheme, okay? Please remember this name, Z-scheme, all right? I know it doesn't look like a Z, okay? It's actually Z if you flip it 90 degrees, like, you know, it's like Z, okay? I always wonder why not N? Like, why not N scheme? Right? But people call it Z-scheme, so, you know, I don't invent this, I just, you know, use it, okay? So this is the Z-scheme. Seriously, I don't make this up. In your book, it says Z-scheme too. Okay, so it's really called Z-scheme. Now, Z-scheme is the actual light reaction that occurs on your photosynthetic microorganism. Okay. Basically, you have two photosystems: photosystem 2, photosystem 1. Okay. Photosystem 2 occurs first, and then photosystem 1. And then the question is, how come we don't call this guy photosystem 1? Why not this photosystem 1 and then that guy photosystem 2? Because, yeah, this guy is discovered first. So, 发现的顺序, okay. All right. Photosystem 1, photosystem 2, they have very different functions and very similar functions. What am I saying? Like your professor is going crazy? No. Okay. So photosystem 2, all right, is where the electron coming from. Okay, so all right, look, on photosystem 2, you have this little thingy called the O2 evolving complex, OEC, O2 evolving complex, or oxygen evolving complex. Okay, now what happens is that in the photosystem 2, you have a reaction center that has a I don't know, chlorophyll, okay? But it has a reaction center. Then this guy is called P680 because it's going to use or absorb light. Okay? So it uses the energy of a 680 nanometer light, okay? Now, when this photosystem or the photocenter, when this P680 gets excited, absorbing light coming down, and it gets excited, so this guy is excited, like excited. All right. When it gets excited, it can then pass the electron. So excited is marked by the little, xing xing up there. I forgot what it's called. Apostrophe? No. Excited state. Okay. So it's the excited 680. Now it can pass that excited electron onto a downstream partner. Okay. And then that guy can pass the electron to the downstream partner, and then keeps on passing, and passing. Okay, so this is just like your electron transfer chain. Okay, just passing electrons. All right. Now, as it's passing electrons, we'll see this in detail a little bit. But what you will get is you will get a proton gradient, just like ETC. Okay. All right. Now, going from this, the middle guy, the middleman here, cytochrome B6F complex. Okay. Will pass the electron onto this thing called the plastocyanin, which is another, it's a protein that carries the electron. We'll see the details in just a bit. Plastocyanin can then throw the electron onto P700. So this is another reaction center that can absorb light at the 700 nanometer light. Okay? Make sense? Right? You absorb another light, you excite this reaction center up to 700 excited, okay? And then that guy can then pass the electron onto, subsequent partners, iron sulfur clusters, essentially reaching this guy. This is called a FD 什么？有没有知道？ Ferredoxin. Okay? Ferredoxin is a soluble one electron carrying protein. So this guy is a protein. Okay? It's a protein electron carrier. Right? Like NADH is a molecular electron carrier. It's like a molecule that carries electrons. But ferredoxin is a protein electron carrier. So inside it, you have iron sulfur cluster. So, but it's soluble. That's the key. It's soluble. Now, ferredoxin, okay, this guy is soluble, and it can then pass the electron onto NADP using this enzyme called the ferredoxin NADP oxidoreductase. Okay, so FNR, I think. Okay, and once it get down to NADP, you generate NADPH. Okay, so the net product is NADPH. And proton gradient. Okay, don't forget the proton gradient. Now, let's take a look at electrons. Okay, so let's take a look at electrons. Now, let's go from the back. Okay, so if your 700 is excited here, and the electron, so this movement, this is all electron movement. So if the electron goes from 700 to this A0 to A1 to blah blah blah, so your P700 will become positively charged. So your 700 loses one electron. So where does your P700 gets the electron from? From plastocyanin. Okay, so the electron is coming from plastocyanin, leaving to this A0 right here. Okay, so electrons balance. The net input is from plastocyanin. Net output is going to this A0 thing. Okay? Makes sense. Now, plastocyanin's electron comes from excited P680. Okay? Now, P680 is right here. So where does P680 gets its electron from? It's coming from water using this OEC thing. Okay? Right? Using the oxygen evolving complex. So obviously what you see here is that the oxygen evolving complex can split water into, essentially hydrogen and O2. All right, this is overall 那个啊. If you can understand this, you don't have to read this, okay? And this, okay? So this is for those of you, 可能听不懂我在讲什么，有没有？你可以回家看一下。One more thing. What you notice here, Z scheme, right? What you see is that the 700 is always higher than the 600. Okay, that is because the scale is this. 刚有没有人在 pay attention to 这个 scale? This is talking about the standard reduction potential. Okay, going from the top being negative, the bottom being positive. Okay, 来 standard reduction potential negative 好还是 positive 好？你要先问我好是什么意思，对不对 ？Okay. Negative more reduced 还是 more oxidized? Typically. 我们说那个 electron transfer 嘛，都是从 positive 到 negative 还 negative 到 positive。negative 到 positive 对不对？所以 negative 比较 reduced 还是比较 oxidized？ reduced. Yeah, reduced. Okay? So negative is typically more reduced going to positive, which becomes oxidized. Right? Okay? When we're talking about energy, usually the more reduced this thing is, the more energy it has. So here is high energy, and here is usually lower energy. Okay? So just keep an eye on that. All right. So that was sort of the energy diagram, and now we're going to look at the physical arrangement of the light reaction components. Okay? So you have photosystem 2, photosystem 1. Okay? And obviously these proteins are going to be on the membrane, just like your ETC. Okay? They're on the membrane. They're membrane-bound proteins. Okay? Now, what you should also notice, okay, is that the oxygen evolving complex, or the water for your electron source, is coming from the lumen. It's not coming from the stroma. This is very important. Okay? This is very important. That the water is in the lumen. Okay? And essentially, what it does is it splits water in the lumen become O2, proton, and electron. So 2H2O becomes O2, 4H plus, 4E minus. Okay? Obviously, electrons go to the P680. Okay, so electrons goes into the electron transport chain kind of, and protons, okay, stays in the lumen. Okay, protons stay in the lumen. Molecular oxygen, because it's a gas, it diffuses. Okay, and the electrons, okay, electrons are going to go through photosystem 2, eventually going to this Q, okay, this Q thingy. Guess what? Q, pretty much the same thing. Q and the Q10, 你知道我讲什么吗？知道哈？可以啦，好不好？就是 Q 嘛，就是那个quinone and quinol 嘛，对不对 ？Okay, so Q can do what? Q can do the Q cycle and then bring in protons from the stroma from the outside. Okay, so the Q reduction part, right? The part where Q is turned into QH2 is going to be right here. Okay, around the stroma. So this is where Q reaction occurs. Just like your complex 1 of your ETC. 听懂我意思吗？就是 complex 1 ETC 我不是说那个 Q 是在那个 membrane 跟那个外面的那个 interface 有没有 ？OK， 那这边一样，就是 Q 呢是要利用外面的 proton 来变成 QH2。OK， 因为 membrane 里面没有 proton。OK， 你要是跟我说 membrane 里面有 proton， 我应该从那2楼跳下去了，好不好？所以 membrane 里面没有 proton 这件事情。OK。Alright, so the proton has to come from stroma, and as QH2 becomes Q again, okay, then the protons get pumped into, guess what? Lumen. Okay, so this is how you generate your proton gradient. Okay, and then your electrons get passed to this thing called the PC, which is plastocyanin, which then can interact with photosystem 1. Absorb another light, and then it goes up and blah 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 blah. Essentially, it goes to ferredoxin. Okay. Now ferredoxin is in the stroma side. Okay, because ferredoxin have to go out and do stuff. Okay, interacting with NADP, make NADPH. Okay, and NADPH can be used for whatever stuff that it's used for. Okay, so this is the spatial organization of your photosynthetic machineries. Right? So okay. You have to know where the proton's coming from. Okay. All right. So this is just another clear view of the Z scheme, okay? And what basically I want to show you is this total 12 photon, okay, per 4 electron, okay? 4 electron equals 2 NAD pH, okay? Because each NAD pH is 2 electrons, okay? Right? Because like if it's only 1 electrons, you have a radical, and NAD pH is not a radical, it's a 2 electron carrier, okay? So we talked about NADP always carries 2 electrons, okay? All right, so let's talk a little bit about photosystem 2, okay? So this is what photosystem 2 looks like. It's a bunch of proteins together, like hooked together. So you see all these ribbons, right? Different color, right? Those are different proteins, different polypeptides. Okay? They come together and form this big complex called photosystem 2. Okay? Inside it, okay, you have this oxygen evolving complex, or OEC. Okay? And the oxygen evolving complex, OEC, it contains these manganese ions. Manganese ions. And I think it's also a, there's also a calcium. Okay, so it's a calcium and then manganese, 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 manganese. Okay? Now, the structure of this is really, for your purpose, not important. Okay, so all you need to know is that your oxygen evolving complex, it contains manganese, and obviously manganese is it's like a metal which can then do electron passing process. Okay? Right? So these manganese ions participate directly in the water splitting reaction, okay? And they do so by changing their redox state, right? And it's not important what the redox state looks like. If you're interested, the detailed mechanism is on page 686 of your textbook, but I bet you anything none of you will be interested, right? Too honest? Okay, whatever. But you know, if you're interested in the the actual chemistry of what people think happens, take a look. Okay, take a look. But for our purpose, we just have to know that the manganese, this whole cluster thing, right? This cluster, it has essentially 5 states. Okay, we usually call them S0, 1, 2, 3, and 4. Every time when it changes state is when you take 1 electron away from it. Okay, so photons used to excite your P680, right? And then 680 gets excited, throws the electron away. Okay, it throws the electron away, and then it becomes P680 positive charge. So then P680 positive will grab an electron from the manganese cluster. Okay, grab an electron and regenerate P680 neutral. Okay, and then you have another wave or light coming in and excite this guy, electron pops away. Okay, this is the same P680. Okay, 同一个哦，不是不同的哦。The same P680, right? And then you're going to grab another electron from the manganese cluster again. So it turns from state S1 to S2, S2 to S3, S3 to S4. Okay, now you took out 4 electrons from that manganese thingy, okay, that reaction center. Then this guy at this point is like, yo, I'm not happy, okay? So what I'm going to do is I'm going to take the electron from water, right? So it's then going to interact with water and split water into oxygen, okay? And then the electrons are going to be stored in the S0 again. And then proton is just, plus proton, okay? 4 proton. Does that make sense? Okay, so it's like not happy, okay? Right, so this is sort of a very, very, very brief description of what happens in the oxygen evolving complex, but the details are in your textbook. All right? So really the net effect of photosystem 2, okay, is that the system in effect stripped out 4 electrons from the 4 hydrogen atoms in the water, in the 2 water molecules, okay? Because you have 2 H2O, right? So essentially you have 4 hydrogen, okay? And you split that into 4 electrons and 4 protons, okay? So basically the net reaction is this. 2 water, okay? Actually, I don't care about water. Yeah, seriously, I don't care about water. What I care about is 4 electrons requires 4 light, okay? 4 light molecules are used to essentially kick out 4 electrons. Okay, so this is what happens in photosystem 2, at least the base part. Okay, and obviously the 4 electrons comes from the 2 water, the 4 hydrogen atom. And then this guy in the lumen, this guy diffuse. The electrons are then transported to your cytochrome B6F, okay? But of course, in the middle, you have transported to Q to this guy. Okay, we'll see that, what happens. Okay, so this is a pictorial view of the photosystem 2, where you have the manganese cluster right here, oxygen evolving complex. And this is your P680 chlorophyll center right here. This is chlorophyll center. And it can pass things onto other, electron accepting partners, okay? Right? And at the end, you get this PQ business, and I'll show you what that PQ is right now, okay? PQ, plastocrenone. It's different from your ubiquinone, okay? Ubiquinone, this Q10, that's what you have in your mitochondria, okay? Q10 because it has 10 isopenoids repeats, okay? Plastocrenone, very similar. It's just that the N right here is 6 to 10, not necessarily 10. Minor detail, okay? But, okay, the key is that it has the same structure. However, you don't have a methyl group right here, okay? So this also different, okay? And, okay, instead of methoxy, here is methyl group. Okay? So the substituents on the ring is different, but it does not change the behavior of this molecule. Remember, the behavior of this molecule is dependent on this part. Whether it's ketone or alcohol. Okay? So plastocrenone, if it accepts electrons, okay, it can turn into plastocrenol. Okay? But the structure really doesn't matter. But you just have to know the different things. Right? PQ and Q10. They are different things. But you turn quinone to quinol. Okay? Initially you have 1, 2, 3, 4 pi electrons, okay, or 4 double bonds. Now you have 1, 2, 3. Right? So that tells you you're doing a reduction, okay? 4 double bonds become 3 double bonds, reduction. Thank you. Okay. All right. So again, Q become QH2. This thing is using a Q cycle. We talked about Q cycle last time. So that's how it brings in the protons. Okay? That's how it brings in the protons. And the Q can then donate the electrons onto your cytochrome B6F, okay, which is a complex. And this complex, what it's going to do is it's going to pass the electron essentially to this PC, okay. Now, the cytochrome B6F complex looks like that, okay. It's not like you're going to remember what it looks like, but it has some hemes and iron sulfur clusters, okay. If you compare it to complex 3, you see some similarities. So you can think of your cytochrome P6B6F complex similar to your complex 3, right? 看起来有点像啊，一点点啦 ，minus 这一坨 ，okay? Right? So cytochrome B6F complex, okay? You have the Q cycle, okay? Q cycle, right? Q cycle, you then bring in your Q cycle and then you generate your PQH2, and then QH2 can then do its business of passing the electrons to, remember Q cycle, when QH2 comes in and then become Q, it does a homolytic cleavage, like 2 electrons come out. 1 electron goes into downstream and the other electron sort of recycle back to do some other stuff, to react with another Q radical or another Q, right? So the electron gets passed down down the protein, eventually reaching this plastocyanin, which has a copper in the middle, Cu. Okay, so this guy is a copper 2 plus, copper plus. Okay, and then plastocyanin pass the electron onto P700, and P700 looks like that. It's 很多 proteins 啦 ，OK， 反正就很多 protein 叠在一起。All right, so photosystem 1, your plastocyanin right here, and here it says that plastocyanin is a small protein containing a copper ion, okay, and the electrons are stored on the plastocyanin, and this is on the lumen side, okay, remember, and plastocyanin is going to come in, and it's going to pass the electron into your photosystem 1. And the reason they can bind is because you have some electrostatic interaction right here. Right? But of course, right? So then when plastocyanin bind to photosystem 1, it can then pass the electrons into your photosystems. Okay? Now this is P700. It has to absorb photons. Okay, so okay, excite that guy, and then you can go into your iron sulfur clusters and down the electron transferring partners. Eventually, you will reach this thing, ferredoxin. Now again, ferredoxin is a soluble electron carrying protein. Okay, so this is already in the stroma now. Okay, so ferredoxin is the net product of photosystem 1. Okay, so after photosystem 1, ferredoxin is reduced, and ferredoxin can then do stuff. Also known as reduce NADP, aka also known as. Okay? All right. Photosystem 1. Okay, so this is a better picture of what it looks like. So you have these sort of resonance transfer, all these resonance transfer, and it's going to pass it down to chlorophyll, the active chlorophyll, and it's going to pass the electron eventually to ferredoxin. Okay, I like this picture better just because it's clear. Okay, and ferredoxin can then be converted into NADPH in the stroma, okay, or cytosulfur cyanobacteria, using another enzyme called the FNR, ferredoxin NADPH or NADP oxidoreductase. Okay, so after all of that, this is the net summary of the light reaction as they occur in a cycloid. Okay, 2 water, 8 light gives you 2 NADPH. Okay? How come 8 light? 为什么需要8道光？一个 electron 1道光 ，NADPH 有几个 electron？2个 electron，然后你有2个 NADPH，so you have 4 electrons 8道光的原因是因为你有2个 photoreaction centers, right? 你有一个680，一个700。So for every electron that gets passed to ferredoxin, you need 2 light. Okay? So make a note. Every ferredoxin, you need 2 photons. 1 photon is 680, the other one is 700. Okay? And in the process, you pumped 12 protons, 不是 photon 哦 protons, okay? 英文正音版有没有 photon 跟 proton? Okay. Alright, so the experimental measurement shows that there are 3 ATP per O2 evolved, and this just so happens to be, recall from ETC, 1 ATP is generated from 4 proton movement back into the matrix, right? So remember when we talked about ETC, it's 4 proton gives you 1 ATP. Same thing here. You have 12 protons that can give you 3 ATP. Okay? And the ATP generation is using, you know, this, 有没有看起来很 familiar? F0 and F1, 有没有? Yeah, ATP synthase. Okay? Okay, so this is ATP synthase. Alright? 可以吗？可以啊齁。所以 photosynthesis light reaction 就是怎样？8道光加上2个水，可以给你4个 NADPH 跟2个NADPH and 3个 ATP。OK， 所以8道光给你2个 NADPH and 3个 ATP。OK， 所以8个 photon 给你2个 NADPH and 3 ATP. Okay, so memorize that, please. Okay, so 8 photons gives you 2 NADPH and 3 ATP. Okay. All right. So if you look at the arrangement of these thylakoid membranes, okay, it turns out it's actually quite interesting that the ATP synthase are always on the non-stacking portion. Okay, so they always face outside. So this is like stroma, and inside is lumen. So ATP synthase obviously always face outside, right? Because if it's in here, it doesn't have space. Okay, so in here, so where the thylakoid, remember we saw that thylakoid is like, 我不是在画大便哦。Okay, so thylakoid membrane, remember how it's like stacks? And like in the stacks here, it's typically more rich in photosystem 2 and cytochrome B6F. Okay, and then you're wondering like how come photosystem 1 is not a lot? Because remember that photosystem 1 is the net, the last one. 我们看我就 confused. So photosystem 2 occurs first, right? So photosystem 2 does not need to touch the stroma, right? 因为 photosystem 2是那个 water splitting 的那个 part 嘛，对不对？ Photosystem 1 spits out ferredoxin, okay? So photosystem 1如果在这边的话，你的 ferredoxin 会卡在中间，合理吧？So photosystem 1 is going to be also facing outside, so that photosystem 1 you can spit out ferredoxin, and then this guy spits out ATP. Okay, so it makes sense, kind of. Okay. All right. So right here. Now, so if we compare the proton movement in different biological systems, in the mitochondria, we have intermembrane space is rich in proton, and when it comes out into the matrix, you generate ATP. So remember at the very beginning I said thylakoid lumen is the same thing as intermembrane space with a lot of protons. Stroma is the same thing as matrix with, you know, ATP generated in the stroma. So these two pictures, this one and this one, you just flip it and then it's the same thing. Okay? Now, if we look at bacteria like E. coli, E. coli's ATP synthesis occurs also on the membrane. However, it does not, because it doesn't have a mitochondria, so ATP synthase actually happens on the cell membrane. So that's E. coli. Okay? the cytosol, this is intermembrane space because E. coli has two membranes, right? It's not just one membrane. Okay? Right? So this is a figure showing you photophosphorylation and the oxidative phosphorylation relationship. Photophosphorylation is essentially the same thing as what we say the photosynthesis machinery. So you're using photosynthesis to generate ATP. Okay? And oxidative phosphorylation is the ETC that we talked about. Okay? Now, it is not a coincidence that they do pretty much the same thing, right? Electron passing, you know, in the end you generate a proton gradient. Proton gradient makes ATP, right? So it turns out, okay, some of the electron carriers can be shared. For example, plastoquinone can be shared. Your cytochrome B6F, complex 3, okay? Remember we saw that the cytochrome B6F complex is very similar to complex 3 in structure. So they're shared. And you can also share cytochrome C6, okay, this other cytochrome, which is an electron carrier, like your plastocyanin, okay? So in the photophosphorylation, in the photosynthesis direction, light is used to split water into electrons and O2, okay? So the electrons get passed in this direction, eventually reaching NADPH. That's photosynthesis. Now, of course, the same bacteria, the same plant or algae at night, you don't have light. What you have to do, you can then use the organics that you stored, like your NADH, okay? You generate NADH from degrading glycogen and that kind of stuff. You generate NADH, and then you use complex 1, okay, to take away the electron from NADH, pass the electron onto the same partners, eventually reaching complex 4, which is cytochrome C oxidase, remember that? Okay, and it's going to convert throw the electrons onto oxygen, and you make water. Okay, 所以这边是 ETC 啊，好不好？这边 ETC 啦 ，right？ 然后这边是 photosynthesis 啊。只是要跟你说，那个小学，我不知道你们有没有啊？我小学啊，老师说晚上不要出去跑步，有没有？为什么？因为你晚上出去跑步的时候，可能什么那个氧气的 level 比较低，那听他骗人，怎么可能差那么多？为什么？晚上对不对？没有光 ，OK，no light at night，OK，废话，不然怎么会是晚上 ？OK， 晚上没有光，所以你的植物要活要干嘛？要做 respiration，OK， 所以你的植物会做呼吸作用，所以这时候你的植物会跟你抢 O2，right？ 所以说晚上不要出去跑步，我是觉得是骗人的啦，可能只是。啊，但早上有光啦，它就做氧气啦，对，你的树就会开始做氧气，有没有？所以你早上去跑步的时候，你的氧气会比较够，最好会差那么多啦。OK， 那有没有可能只是小时候老师要跟你讲说晚上不要出去玩，会怕被绑架之类的？有没有可能？有可能嘛。然后用一个好像很科学的理由告诉你，有没有？所以晚上，晚上。你的树因为没有光 ，OK，no light for the plants, so the plant has to do respiration, okay? So respiration, this thing is respiration, 可以哈 ，right？ 所以就这个意思啊 ，right？ There's this thing called cyclic electron flow, okay? Cyclic electron flow. This happens when, okay? This happens when your NADPH is too much. You have too much NADPH, okay? Like, too much NADPH. I don't know what to do, okay? I don't need to make more NADH, okay? NADPH. If your NADPH is too much, that means that your ferredoxin also too much, okay? So, you know, remember that ferredoxin is like NADP become NADPH. If this guy is too much, this guy is little, okay? So then this doesn't happen. So you have a bunch of ferredoxin. What do you do? Okay, what do you do? Turns out, your photosystem 1 can do this really crazy thing of, let's say, okay, I throw the electrons from ferredoxin back to the cytochrome B6F complex. Okay? Just like 我 electron 太多，我不要了，可以吗？然后我再把它丢回去 cytochrome B6F. Okay? Too much electron, throw some back. Okay? And then cytochrome B6F, throw it onto PC. PC, throw it onto P700. Okay? And then this guy gets excited again. Okay? And in the process, what happens is that you are going to transfer 2 protons from the stroma into the lumen. Okay? So cyclic electron flow is used to generate ATP without net electron movement. Okay? So you don't need to split water or anything else. All you have to do is just shine light and then keep on doing this thing, and then you're just pumping protons into the lumen, and then when the protons come out, you generate ATP. Okay, now, so for every electron, so every 1 electron, you move 2 protons. We said we need 1 ATP, we need 4 protons. So we need what? 2 electrons for 1 ATP. Right? 2 electrons equals 2 photons. Okay? Right? So, 各位, just remember this. Okay? In photosynthesis, every time you're moving 1 electron, you need to have 1 photon. Right? So electron, photon. 1 electron, 1 photon. Okay? Now, if you're doing photosystem, if you need to use photosystem 2 and photosystem 1, then it's 1 electron, 2 photons. Make sense? 懂我意思哈？所以如果你要从 photosystem 就是那是什么 2 是不是 photosystem 2打一个那个 electron 过来，你就需要2道光 ，OK？ 你需要2个 light。但是如果你只是做 cyclic electron flow 的话，你只需要1个 light， 然后让它就是继续跑这样 ，OK？ So very similar to cyclic electron flow are these purple bacteria. These purple bacteria, they don't do the Z scheme. Okay, they don't have photosystem 1 and photosystem 2. What they have is one single photosystem. Okay, they have one single photosystem, and this system is somewhat similar to the photosystem 1. Okay, it's similar to photosystem 1 such that you do not do any water splitting. Okay, all you do is you pump electron and the electron moves around, and as a result you generate a proton gradient. Okay, so that's very similar to photosystem 1. Okay, and there's a reaction center. Okay, and there's also a membrane-bound cytochrome complex. Okay, so you have 2 large enzyme complexes involved in this particular type of processes. Okay, so what happens is that when you have a reaction center right here, photocenter, and it says it's 870, so that means that it absorbs at the 870 nanometers, which is in what region? Do you guys know? I mean, of course you should know. 870 is in what EM wave region? Huh? 不是 UV 哦。UV 的波长比较短，对不对？因为 UV 的能量比较大，比可见光的能量要小的是 IR, right? Infrared, right? So your reaction center for 870, so this guy can sort of absorb near infrared wavelength of light. Okay, so it behaves differently from our photosystem 2 and photosystem 1 because it doesn't absorb in the visible, it actually absorbs in near infrared. Right? But whatever, that's just a detail. Okay. All right. So what you notice is here you don't have water in this entire process. So water is not split. Okay. So it's really more of a cyclic electron flow. Okay. So it's a process that is used to generate a proton gradient. Right. So when you have an excitation of light that hits the reaction center, again, you excite your reaction center to a higher excited state. Okay, the electron, one of the electrons get excited to the excited state. And now you can do electron transfer. Okay, you transfer electron from this guy excited state to this guy's excited state. Right? So you pass the electron to this guy, and this guy can then pass that electron to Q. Okay, and guess what? You have this thing where Q can then grab in the protons and pass that electron to cytochrome BC1 complex. You don't have to remember the names, okay? You just have to know like it's a cytochrome complex, okay? And once it passes the electrons into the cytochrome complex, so it's going to pass the electrons around, and then you'll pass it to another cytochrome protein, which then can carry the electron back to the reaction center. Okay, so this is the energy diagram. Okay, this is the energy diagram. If you look at the sort of a pictorial view of it, so what happens is that you have the reaction center right here, and once it absorbs light, it passes the electron inside, okay, gradually reaching Q, okay, and you're going to grab the protons from your cytosol, okay, similar to your Q cycle thingy, okay, and you'll grab that proton and then pass the electrons, okay, from quinol, okay, remember Q is quinone and QH2 is quinol, and quinol can then pass the electrons into the cytochrome complex, and then the protons are then ejected into the periplasmic space, okay. This is called periplasmic space because this guy is a bacteria. Thank you. Okay, so it's a bacteria, so it's a periplasmic space. It does not have lumen, okay, like what we call the chloroplast guys. Do you guys even understand what I'm trying to say? Okay, maybe okay. Right. So the protons are then in the periplasmic space, right? And your electrons, once it goes onto the cytochrome complex, it can go to this other thing called cytochrome C, and then cytochrome C can then pass the electron back to A7D. Okay, so we're good. All right, so electrons are happy, and then you increase the concentration of proton, and you have a proton gradient, and proton gradient gives you a proton motive force, and that guy, when it comes back into the cytosol, you generate ATP, okay? So repeatedly in biology, you see the same process over and over, okay? So it's not a coincidence that these things are this way. It's like evolution. They kind of just did it this way, okay? Okay, so this is cyclic electron flow that occurs in the purple bacterial photosynthesis, okay? This is the reaction, the photoreaction center of photobacterial photosynthesis. So, you know, again, just to show you, it's a bunch of proteins together, okay? The reaction center, it has a bacterial chlorophyll. Right? It's not the chlorophyll A or chlorophyll B that we use for the Z scheme. Why? I mean, come on. In the Z scheme, it absorbs 680 and 700, right? So the fact that this thing absorbs energy at like 870, probably a different molecule. Maybe? Yeah, okay? So it has a different molecule that has a maximum absorbance at 870 nanometers. Therefore, it's called 870, okay? Turns out this is actually an example of an oxygenic photosynthesis, okay? Do you guys know what the word an oxygenic means? Remember last time we talked about the Z scheme happens for oxygenic photosynthesis. Okay? What does oxygenic mean? Genic means it produces something, right? It generates oxy, probably oxygen. Okay, so oxygenic means that it makes oxygen. Okay, so anoxygenic means that it does not make oxygen, and typically what it also means that is that these bacteria can be anaerobic bacteria. Do you guys know what that means? What that is? Anaerobic bacteria. So bacteria that grows in the absence of oxygen. Okay, so their growth is not coupled to oxygen. Okay, so they can grow in environments where you don't have a supply of oxygen, but yet they can still do photosynthesis. Okay, but it's just that their photosynthesis does not produce you electrons. Their photosynthesis can only generate ATP. Okay, but that's already pretty cool. Okay, that's a very good process. Right. So these microbes can then use the ATP generated to then strip the electrons from hydrogen sulfide. Okay, so that's where you get the electrons for generating NADPH from, but the electrons is not from water, right? So when you want to split hydrogen sulfide, what you can use is you can use the ATP generated in this cyclic electron flow, okay? Because this guy can give you ATP, and then ATP is equal to energy, and then you can then use this energy to split your hydrogen sulfide into sulfate. Remember at the very beginning we saw the different bacteria, different carbon source, electron source, that kind of stuff. So then you can split hydrogen sulfide into other stuff, okay? And it becomes oxidized as sulfate, okay? And in some processes you can also use hydrogen gas as the electron source. Hydrogen gas is actually a very useful electron source because this guy can directly be split into 2 protons plus 2 electrons. Right, so hydrogen is also very useful. Right? Okay, so again, we talked about cyanobacteria, okay, which is same thing as saying plants. So this guy you should know very well. This guy, okay, you should know like the Z-scheme, what happens in the Z-scheme, what's the stoichiometry of the Z-scheme, okay, what the proteins look like. No, you don't have to know what the proteins look like, but you have to know that most of them are protein complexes, okay, and you need to know, ferredoxin is higher energy than NADPH, for example, okay, and you also got these purple sulfur bacteria that can grow using a cyclic electron flow, and cyclic electron flow which then allows it to generate ATP, and it can then use that ATP to deal with the hydrogen sulfide, okay, or using H2 as the electron source, right? The purple guys. These are the purple guys, right? So just it really is for you to understand that there's a lot of different photosynthetic organisms out there, okay, not just plants, okay, even though most of us, when we hear photosynthesis, we think of plants, but in reality, it's a lot more. Now, this is actually another very interesting example of a photosynthesis, okay. It's also kind of like a photosynthesis, but it's really powerful, okay. This thing happens in some archaea. Okay, so not bacteria, not eukaryotes. Okay, so it's archaea. Okay, do you guys know what archaeas are? 就是它跟 bacteria 不一样嘛，它的 cell wall 也跟 bacteria 不一样，它其实有很多东西都跟 bacteria 不一样。OK， 那但是它们 most likely most of the time 也都是微生物啦。OK， 所以不是什么大型的 organism, right? Archaea. And these archaea, they have one single protein. Okay, they have one single protein that can be used to pump, 这个 positive is right here, okay, it's on the top, okay. They can use one single protein to pump protons across the membrane, okay. Previously, when we saw cyclic electron flow to pump protons, you still have to use a protein complex, okay. And you have to, for example, in this case, you have one big complex and another complex, okay. So you have a system here. However, in archaea, it's actually very unique because they can only use one single protein and then they can pump protons into the intermembrane space or the periplasmic space. Okay? And this protein is called the bacteriodopsin. Okay? Even though it says bacterial, it's not bacteria. Okay? Just don't get confused. Okay? This protein is called bacteriodopsin. And it's a protein that contains retinal, okay? It's an aldehyde derivative of vitamin A, whatever, okay? It's just, as long as you understand it's coming from vitamin A, And vitamin A is what? It's colored, right? Because you can actually see vitamin A. Vitamin A can do what? It can absorb light. Okay, you can absorb light. So the functional part, the part that absorbs light in bacterial rhodopsin is your retinal. Okay, is your this vitamin A guy, and when it absorbs light, you will be able to pump protons across the membrane. Okay, and the way it works is kind of like this. It's one single protein, so you can see it's one protein. But of course, it's a membrane-bound protein because in order to pump the proton through the membrane, it itself has to be integral membrane protein. You know, it has to have many transmembrane domains. Okay, and what it's going to do is, this is the cytosol. This is the external medium, or just the part where you want to keep a higher concentration of protons. Okay, what happens is that your light will hit this retinal. Okay, I'll show you the detailed mechanism in the next slide. But essentially, what happens is that you will trigger a conformation change, okay, a conformational change to the retinal, and then, okay, and then it will cause the pKa to drop, okay, such that it can pass a proton to the aspartic acid, and then you pass the proton downstream until where you get to a water right here. There's water right here. You'll see it more clearly in the next slide. But once you get it to here, and then you can sort of kick out the proton into here, right? And all of this is due to a conformational change of your retinal. Okay? So it looks like this. This is your retinal. Okay? It's a conjugated molecule, right? It has alternating double bonds. Okay? And it's attached to a lysine, okay? And this attachment is the shift base formation that we previously talked about in, do you guys remember which protein, which enzyme in glycolysis has a mechanism that uses shift base? 有人记得吗？ Glycolysis 里面10个 enzyme 有没有？我们有讲到其中一个 enzyme。 它的催化机制里面会形成一个 shift base。第4个，第一个什么 hexokinase 嘛，然后 hexokinase 后面是那个 isomerase 再一个 kinase， 是第4个没错，是那个 aldolase 有没有？那个 aldolase 它不是有2种吗？一种不是用那个 zinc 的，然后另外一种就是用 shift base 的 ，OK？ 所以again 要跟你们讲说 shift base 在 biology 里面常出现啊，所以对这个名词至少不要太陌生 ，OK？In case 你们 later on 有做一些相关的 work, okay? All right, so guess what? You have a retinal and it comes to this enzyme and you make a shift base, okay? Out of it, and shift base essentially means this guy, okay? And typically your shift base have a high pKa, typically, okay? So this guy is relatively stable, no problem, okay? So it's relatively stable and nothing really happens, okay? Now, once you expose light, my weird drawing of the sun, okay? Once you expose it to the light, and you will trigger a conformational change to the retinal, so you actually turn it from trans to cis, kind of, okay? And you will lower the pKa of the shift base, okay? And lowering the pKa means that the proton can be deprotonated, okay? So the proton gets deprotonated and you pass it down to a neighboring aspartic acid, which will then sort of trigger a series of proton movement from water. Eventually you get down to the pKa of the proton release complex. Okay, so this part is the proton release complex, and the pKa is lowered. Okay, and because it's lowered, okay, lower pKa means that your proton can leave the molecule. Okay, leave as a proton. Okay, and of course this conformational change is not permanent. Okay, it's not permanent, so it's going to grab another proton onto your shift base, and then you essentially regenerate this guy. Okay? But for your purpose, really, just as long as you can understand, bacterial rhodopsin is guess what? One protein. Okay, it's one polypeptide type of enzyme, and inside it you have a prosthetic group that looks like a vitamin A, and yeah, okay, it can pump protons. Right? So a while ago, I think it was like maybe 6 years ago, there was a group that was wondering, this mechanism is very nice, okay? Because, for example, in my work, a lot of times when we design synthetic pathways, we need to have ATP, okay? 就是像我做research 的时候，我们有时候在设计一些合成的代谢途径嘛 ，OK？ 那为什么要做合成代谢途径？很多原因啦 ，OK？ 可能生产一些生殖相关的东西啦，或者是你想要去做让这个 cell 有一个很奇怪的function，然后可能没什么用，但是看起来好像很厉害，这样也不错啦。哎，那有的时候会 ATP short，right？Make sense。Okay？Sometimes 我的 pathway 可能 ATP 不够，那你其实就可以用这个 complex 来去让 E. coli 变成 photosynthetic， 就是说我让 E. coli 去表达这个 protein，OK？ 让它表达这个 protein。那它如果可以成功的表达这个 protein 在这边的话，那我是不是对 E. coli 照一下光，那 E. coli 就可以 pump proton。那 E. coli 如果可以 pump proton 到它的这个 periplasmic space 的话，那 E. coli 自己有 ATP synthase 嘛，那当 proton 再回去的时候，它就又可以做 ATP， 所以我就可以做 extra 的 ATP。所以大概几年前有一篇 paper， 它很好玩，他们就拿这个bacterial rhodopsin 放到 E. coli 里 ，OK， 但是他们我忘了，他们应该是没有正确的证明出它可以制造 ATP， 但是它确实可以证明它有制造出一个 proton gradient，OK， 他们怎么 prove？ 他就可以看那个 E. coli 的那个毛有没有 ？E. coli 不是会转吗？那个转动的 action 就是靠 proton gradient 在转 ，OK， 所以他就可以看到说，有这个 bacterial rhodopsin，OK， 然后他拿那个光去照那个 E. coli， 然后 E. coli 就会跑来跑去，然后光一关掉 ，E. coli 就停了。Okay， 所以用那样子去测说这个东西有没有 functionally expressed，OK？ 所以you can actually use bacterial rhodopsin 去做一些很有趣的东西。Alright, so that's all I have for photosynthesis. So very briefly talk about, because this figure is in your book. It's called the artificial photosynthesis. So now I don't think I have to convince you guys that energy is one of the most important problem in the 21st century. Okay, so in this century, energy has to be addressed. Okay, because we just even though like 台湾不是什么少子化吗 ？OK， 但是真的啦 ，believe it or not， 整个地球的 population 还是在成长 ，OK， 正成长，所以人口成长，你需要消耗的资源也会要跟着成长 ，OK。However， 我们的资源不是有限，我们资源是有限的嘛，不是说那么好像就是 forever 都会有 additional 的 supply。So then we need to really think about source of energy, okay? And like I said previously, in the future, your energy, the best energy has to be light energy, okay? It has to be photo energy, okay? I mean, of course, nuclear energy is nice, but it's coupled to danger, okay? You have risks. So nuclear energy is not suitable for every place. But eventually, we have to become sustainable through utilization of light, okay? And now, we know that splitting water, okay, water splitting using electrolysis, okay, using electricity is doable, okay? We can do that. However, where do you get the electricity from? Okay, so it's a cycle. So it turns out a lot of people are interested in photosynthesis, okay, because photosynthesis can do this water splitting naturally, and we wouldn't say it's super efficient, but it's not too bad, okay? The efficiency for plants to split water, it's pretty neat, okay? It's pretty nice. So there's a lot of people doing research trying to create artificial photosynthesis. So photosynthesis inspired photocatalyst design, okay? So Here's an example in the book where this is a very simple solar cell, okay, that can be used to generate hydrogen gas, okay? So you have water in the system. You have water in the system, and you have these dyes that can absorb light, okay? And then you have some catalysts, okay? So these are essentially catalysts that will absorb light, okay, and use that energy to then be able to split water, okay? So when water is split, you generate oxygen gas, O2 gas, which can leave the system. And what you can then do is you split water. Remember we said you get O2 and 4 protons and 4 electrons. The electrons are going to be conducted, okay? You have essentially wires, okay? Electron will get conducted away, while protons essentially gets trapped here, right? And then when you combine electrons and protons at the end, okay, you will be able to generate hydrogen gas, okay? So unlike the natural photosynthesis, your electrons ends up at NADPH. Here, your electrons sort of recombine with the protons, and you can generate hydrogen gas, okay? And hydrogen gas can then be used to supply clean energy and that kind of stuff, okay? So, all right, so that's the artificial photosynthesis. So a lot of people are trying to use the photosynthesis inspired. And why do we call this like photosynthesis inspired or artificial photosynthesis? Because here you're using manganese as well, okay? Just like your real photosynthesis, you also have that manganese thingy going on, and here you also have that, okay? So that's it.